So, hi, everybody. Welcome. Thank you for joining us today. My name is Elena Lazarus, and I'm the research coordinator for the Water and Life Interface Institute, also known as WALI. And I'm also your moderator for today's talk. Today is the second talk in our series, Anhydrobiosis Across Organism Scales and Disciplines. Our speaker today is Jenna Ekwealor. Jenna is an assistant professor of biology at San Francisco State University. Prior to joining San Francisco State University, she completed a PhD in integrative biology at the University of California, Berkeley. At SFSU, her research is in the Moss Eco Evo Physio MEEP lab, which studies integrates plant biology, phylogenetics, and evolutionary genetics to understand the evolution of plant life history and adaptations to life on land, with a focus also on stress and response to stress mechanisms. If you have questions for Jenna today during or after her talk, please write them into the Q&A box, kind of towards the bottom center of your screen on your control panel on Zoom. We'll have about 10 minutes for Q&A session after the talk concludes, and we'll go over all the questions that come in both during and after her talk at the end of the session. So with that, I'm going to pass the mic to our speaker today. And Jenna, thank you for joining us, and please feel free to start your talk.
Thank you so much. Thank you for having me. Thank you all for coming. I'm really excited to be here to talk to you all. Excited about the Q&A. Of course, if there are issues with my audio or anything, feel free to, I guess, reach out to Elena. But yeah, so I'm going to be talking about, harnessing the sun, the beneficial role of ultraviolet radiation in desert moss desiccation tolerance. And while in these projects, I did learn a few things and answered some questions. I think a lot of what this talk is about is sort of lessons learned about how organisms have adapted to their environment, even the things that we think of as stressors. They may actually be kind of another resource for that organism. So I'll talk about what I mean by that. But before we get into the projects, I want us to think about my overarching curiosity, and go back hundreds of millions of years and think about those first plants that left the freshwater habitat that they evolved in and colonized land for the first time. Think about what those conditions would have been like. They're suddenly exposed to air and have to grapple with drying out. Or trying not to dry out potentially. And also they'd be exposed to a lot of really intense sunlight without any sort of water filter. And also conditions were harsher back then with high levels of ultraviolet radiation in the atmosphere and really intense sun. So these are the things that sort of drive my research overall, even though I, get sort of narrow at points. But really what I'm interested in is, how do plants survive the irradiating and desiccating conditions of life on land? Because to this day, those conditions are still there for plants. They've just evolved different ways to handle them. And I do this by studying mosses. Now, mosses you might not associate with harsh irradiating and desiccating conditions. You might be picturing a habitat like this. This is the Hoh rainforest in Olympic National Park in Washington State. It is, of course, very mossy. Mosses love it there. Low light, cool conditions, very moist. There are a lot of mosses, it's true. But mosses are actually quite abundant in habitats like this, especially those that are sort of healthy and not terribly degraded. They're just a little harder to see. So many of the mosses live on the soil in communities like this, and they spend much of the year looking dry and brown or even black and kind of dead. People think of them as looking dead. Here in these photos, the mosses are the black and silver sort of spots you see everywhere. And this top left one, among the cyanobacteria, and here making a whole carpet, and here in a pretty mixed community. These are all photos when the mosses are dry. When they're hydrated, they sort of come to life, change color, and I think are quite beautiful. This is from a community in southwest Idaho. So mosses that live on the soil in drylands are members of what are known as biological soil crusts. They've been called the living skin of the earth by other researchers. And there are communities of bryophytes such as mosses, lichens, fungi, cyanobacteria, and other microorganisms, tardigrades, living on the soil surface, the top centimeter or so, in dryland, so deserts and arid and semi-arid habitats around the world. They perform critical ecological functions. In addition to physically holding the soil together, like you see in this picture, and through that, they reduce erosion and soil nutrient loss by wind and water erosion. And these are habitats that are typically nutrient poor to begin with. So that's very important. They increase soil fertility, again, nutrient poor, and water infiltration, very important in a dry habitat. They facilitate germination of native seeds and actually reduce germination of non-native exotic seeds, which is kind of amazing. So they're important communities, and mosses are an important member in many biocrust communities around the world. But they're in deserts and drylands, and so these organisms don't get a lot of water, right? And they spend much of the year without any water. And so they have this trait known as a suite of traits, really, known as desiccation tolerance. They're able to dry out completely, losing virtually all of their cellular water, and go completely metabolically inactive for months at a time, or as long as it takes until water is available again and they can rehydrate. So this is two different clumps of the same species of moss, hydrated and dry. So you can see how remarkable that difference is. And you don't have to just look at photos. I have this video. This is the moss Centrichia cananervis. Centrichia is common in drylands around the world, and this species common in Mojave Desert, where my research takes place. And this video is not a time lapse. Okay, so this is just how fast it is. They were dry in the beginning. I spray some water on them, and you can see the leaves opening up. They turn green before our eyes. And we know from physiological studies that they begin photosynthesizing immediately. They begin repair immediately. And they're very, very fast at this. They have to be, because those rainfall events, precipitation events, could be really short in a desert habitat. So they have to be really quick at this ability. So most of my research is in this group Centrichia, a genus of about 100 species, and many of its members are dryland specialists. But it's also interesting beyond that, because although many are in deserts and drylands, there's a lot of diversity. So there's a lot of different habitats within the group. And I study this group from these different angles: ecology, physiology, phylogenetics, and genomics, to try and answer this same question of how do they do it? How do they survive in these habitats? So I try to integrate these things, which sometimes means that I don't necessarily get all the answers in any one category, but maybe some sort of synthetic answer that I'm looking for. Okay, so for today's talk, I actually gonna cover bits of a few different projects that I think connect really nicely and kind of led to where I'm at today and what I want to do next. So first, I'll talk about hypolithic moss that lives under rocks. A photobiology project that I went into next. So a little bit of the stress physiology work I've done, and future research where I think this is heading and what I want to check out next. Hypolithic moss. So this project actually started kind of on accident. My collaborator, Dr. Kirsten Fisher, and I were in the Mojave Desert, a high elevation site collecting samples for a different sort of population genetics project. And down here, my collaborator was admiring these beautiful white milky quartz rocks that are pretty common here. It's a quartz outcrop in this wash, and there's a lot of this quartz. And she picked one up and noticed there was moss growing under it, even though there wasn't a ton. There's a lot of moss in the site, but as you can see in this picture, there's a bit of moss here, but it sort of seems specifically under that rock too. So we started picking up more rocks and checking, and there was moss under many of these quartz rocks. This led us into a project that involved some students that helped a lot. Jose Medina, from a former high school student, and Caitlin Millette, who did a lot of microscopy work for this. But before we really knew what to do, we went to the literature and searched everything we could about mosses that live under rocks, especially in the desert. And I found really only a single study that focused on mosses that live under rocks. And it actually wasn't even a study. It was like a paragraph in the Bryologist. There was a sort of report that these researchers had found moss under crystalline crystal in the Mojave Desert, a different part of the Mojave Desert. But there was no sort of follow-up study, no information about that habitat. And the only other reports of moss under rocks I could find was in Antarctica, and they were also sort of an aside. So we were curious, what's this is this a real habitat? Is this a fluke? What's going on here? So we set out to answer some questions and figure that out. First, is the hypolithic microclimate different than that of the soil surface? So if you notice in this photo, the mosses look different. At this stage, maybe we didn't know if it was different species or what was going on, but there's this dark color around and green underneath. And so I thought maybe there's something different under there. And do the species that occur under the rocks and next to the rocks differ? Are they different species composition? So first, looking at the species composition question, we compared the soil surface and hypolithic community composition for 18 quartz rocks at this site. And the major players were the Centrichia cananervis. This is a dominant species at this site. This is the one we were sampling, so this was no surprise. And also Tortula inermis, a little bit more of a surprise. This is a desert species, a Mojave species, but it doesn't typically occur at this site, or we didn't think so. We also found some Bryum argentium, which is a cosmopolitan species. You can find it everywhere, but in deserts, it's in pretty low abundance, but it's there. And so we counted number of rock species, where species occurred in these 18 rocks, if they occurred in the soil surface or in the hypolith. And we found for Centrichia cananervis, there were twice as many. It was twice as common in the soil surface as it was in the hypolith habitat. So there's 2-to-1 ratio. Now, Tortula inermis, these boxes are scaled to their abundance in the community. Tortula inermis was much less common overall, and it had the opposite pattern. There was 1-to-4 ratio. So it 4 times as common in the hypolithic habitat than it was in the soil surface habitat. It was pretty rare in the soil surface. So that there explained why we did not think it occurred at this site. It's mostly under rocks, and we had never looked there before. Bryum argentium, as I said, is super rare, and so we found only 1 sample, and it was on the soil surface. So the rest of this, I'll just talk about those first 2. So yeah, from here we could conclude that community composition does differ between the soil surface and the hypolith. There's a different ratio and different proportion of species on each habitat. But So we wanted then, okay, what explains this? Is there something different with the microclimate? So to answer this question, we set out temperature and relative humidity data loggers for 5 months that collected hourly. And here on this graph, I'm going to show you those data, but it is the difference. So it was a paired design. Each rock had one sensor on the soil surface and one under the rock. And what I'm plotting here is the difference in those. So when the line is above the zero, the hypolith is cooler. And when it's below the zero line, the hypolith is warmer. I've also color coded the lines. When they're light blue, it's like daytime hours, and dark blue, nighttime hours. And it goes over these 5 months. All right, so immediately you can see there's a lot of light blue above the line. So there's a lot of time and a big difference in which the hypolith is cooler, especially during the day. Now, this is important for potentially important. It's at least meaningful for a desert where it can get very hot. Under these quartz rocks, mosses are getting at least up to 15 degrees Celsius degrees cooler under the rocks compared to the soil surface next to it. And then at night, we see kind of the opposite, less magnitude, but where the lines are dark blue here, at night, the hypolith is actually warmer than it is on the soil surface. This continues, and then right around here, November and December, there are these two periods where there's not this day-night pattern happening. And during those, it's consistently warmer under the hypolith, a small amount, but consistently so. Those are two snowfall events where everything was covered in snow. This is a high elevation Mojave Desert site, so they do get snow. This was a particularly snowy time. There were two big storms. And during the entire snowstorm, the quartz provided a bit of warmth for under the rock. And this pattern is particularly strong here in September and October, where it's still quite hot and becomes less so, but still apparent through the winter. So what we're seeing here is a buffering effect. When it's hot on the soil surface, the hypolith is a bit cooler, and when it's cold on the soil surface, the hypolith is a bit warmer. So overall, it reduces the extremes for the plants that live under there. Now, another important factor is water, as we all know at Wally. As a proxy sort of for water availability, we measured relative humidity. It can work as a proxy for how long a plant might be able to stay hydrated. And so we did in the same thing, measured in this paired design. And again, I'm showing the difference where above the zero line, the hypolithic habitat is more humid, and below it's less humid. So I'm going to show this in steps here. So first, through these warm, sort of late summer months, we again see a diurnal effect where during the day, there's more humidity in the hypolith, and at night, less. And it's pretty consistent. Then there's a big switch. First of all, right around the zero, you can see those two snowfall events again, where there's really no difference between the soil surface and the hypolith. But the rest of the line is completely above the zero, where day and night, it's more humid under the quartz rock. So this is important. What this tells, I mean, overall, there's a humidity benefit under the quartz rock, and particularly when it matters. So these plants are poikilohydric, desiccation tolerant. They dry out when it's dry. And so they grow mostly in the winter when water is available, rain or melted snow. And so this time when it's more humid under the rock is the time they're more likely to be able to use that humidity because they're hydrated and they can photosynthesize. Likewise, in the fall, we don't know if the plants were hydrated during this period. It's always possible due to condensation or dew. But if they were, that humidity would be particularly important during the day when they could do photosynthesis. So it seems that there is a benefit. They're perhaps not drying out as quickly, or again, maybe something about the quartz ability to stay cold and helps water condense. We're not sure, but some sort of benefit with humidity under the quartz. Another component of the microhabitat, though, is light, right? So these are photosynthesizing plants, and these rocks are all this white color and do not look particularly translucent. So we were very curious if light was getting through. There's some possibility that it wasn't, and the plants, there's other plants that are able to do something like this because of connections to plants that do see the light. For example, strawberries with runners and things. Now, I don't think that's known to occur in mosses, but we had no idea. So we first went to the literature and found a geology study that actually measured light transmission through Mojave milky quartz as a function of the rock's thickness. So the equation that the Beer's law equation that we can use to estimate the amount of 650 nanometer light that transmits through is here. And we did this for all of the rocks in the site. And on average, we found 1%, 1.2% of 650 nanometer light transmits through our sort of average quartz rock. On that note, the quartz that harbored mosses ranged from, I want to say 1 to 4 centimeters, and our average was about 2.5 centimeters. We found that if they were too small, thinner than that, they didn't have moss, and if they were too big, they didn't have moss. Probably the thicker ones didn't get enough light is our guess. But so very, very low light in the hypolithic microhabitat. Now mosses are low light plants. They don't need a lot of light, but still this was quite low. And this is a desert where there's a lot of light availability. So at this stage, we're wondering if this is not a benefit for the plants. Another thing that we noticed was that the light, this is also from the Schlesinger et al. Sort of optical geology paper. And they noted that the light that transmits through is not uniform with respect to wavelength. And here we had percent transmission on the Y and different wavelengths that transmit through on the x-axis. And you can see there's a slight but existing increase in the transmittance line. And what that means is that longer wavelengths, so red and far red, transmit at a higher proportion than small wavelengths, including ultraviolet radiation. So they're not only getting lower light overall, they're getting a different light quality as well, a different proportion of wavelengths. And that may be meaningful. And so I became curious about this lower proportion of ultraviolet radiation in particular. And I'll get more into that. Also, the plants looked different, as I noted. They appeared longer, like larger plants under the hypoliths. So I measured that on 349 shoots. And here you can see this is in millimeters. The hypolithic shoots were about 62% longer on average than the soil surface ones. And here's a picture of just sort of a representative shoot from, this is Centrichia cananervis, from the soil surface and the hypolith. That's a particularly long hypolithic shoot, but it's within the range. At this point, you might also be noticing the color is different, but I'll get back to that. So Is the hypolithic microclimate different? Yes, there's a temperature buffering, higher humidity most of the time, very low light, and lower proportion of ultraviolet radiation. Do the moss species composition or growth characteristics differ? Yes, Tortula inermis is much more abundant as a hypolith, and we think that this may be a refuge from cold. Um, that buffering situation. So Tortula inermis, as I mentioned, is a Mojave Desert species, but it typically occurs down the mountain in hotter, drier conditions. So when we found it sort of hiding out under the quartz, it was a little bit of a conundrum until we thought about those snowfall events and the winter in general being cold. So we think it's possible that this species is less tolerant to cold and is only able to live this high up the mountain in these microhabitats where they're a little bit more protected from the cold. This project was a lot of fun. It was a side quest that we didn't really plan, and published it in 2020, and it kind of had a little bit of a news heyday because I think in 2020 people were really just desperate for like a feel-good nature story. If you remember what 2020 was like. So that was really, really fun. And hopefully I this week I could use a feel-good nature story too. So it's good to be talking about it again. Okay, so as I said, you may have been noticed the coloration is different. This one on the soil surface is very typical. You saw those early pictures. It's usually got this dark brown coloration. But the hypoliths were super green, and the Canonervis, the same species. And this got me thinking with especially considering that transmission as a function of wavelength data. Is there a UV sunscreen in Centrica canonervis that possibly is not induced to the same extent in that hypolithic habitat? So this is what set up my next projects. I became sort of extremely curious about this question. Fundamentally, a very simple question: why are they brown? But it ended up being a lot more complex than that. So this leads into the next section where I get into photobiology. As I mentioned, I started this really wondering, why are they brown? And does UV radiation induce the brown coloration in this species? And how I ended up wondering too, how does UV radiation play into their desiccation tolerance, or does it at all? And I'll explain what I mean by that when I get to that question. But first, this project is a very integrative one. I combined field and laboratory experiments, and it sort of checks these three boxes on my research program agenda. So I had these field treatments for a year, which I'll explain, and also a growth chamber experiment in which I filtered out UV radiation from Centrica canonervis in a natural setting. Then I took all these samples and did stuff in the lab and compared their outcomes. And it was a lot. I'll mostly focus on this part where I monitor their chlorophyll fluorescence over 8 days, but also a bit about here where I look at pigments and antioxidants. So I'll talk about those. Again, I had wonderful students working with me on this. Satuda and Alexander did a lot of the chlorophyll fluorescence with me. And that was a lot of samples to process. So it was really fun to work with them. I did this project also in the Mojave Desert, a different site, a little bit lower elevation, does get occasional snow, but much less frequently. In the Granite Mountains, sort of near Baker, California, if you know the area. And here is what Centrica canonervis typically looks like in this site. It's a very sort of gravelly, granitic soil, and all this black is Centricia nervis growing out in the exposed open sunlight. And for this experiment, I set up 20 UV filtering windows, these acrylic windows that filter out UVB in particular, 100% of UVB. And in a paired design to control for the effects of the window itself, I did these UV transmitting windows that transmit most of it through. This is like what's used in tanning beds and reptile enclosures and things. And I also, for the rest of these data, collected a third totally unmanipulated site reference just to see what's sort of normal and see if there was an effect of the window itself. And so I had 20 triplets like this, and I left them in place for a year and then collected at the end. So the first thing I was curious about was this color. I wanted to know, did UV radiation induce the brown coloration? Is it like a sunscreen? Well, is it like a suntan at this stage? And so here are some shoots from each of those, just a very tiny subset, but they're fairly representative. The UV filtered looked very green, whereas the site reference looked very typical, brown, dark brown. And interestingly, the UV transmitted windows, which should have been the control, they transmit all the UV, or supposed to, kind of looked intermediate. They had a brown coloration, but were a bit greener. So I tried to quantify this. I took photos of lots and lots and lots of shoots, and they were RG, red, green, blue pixels, RGB, using just the shoots themselves, not the background in the photos. I quantified the proportion of green pixels in each of them. And here, this teal is the UV filtered plants, the greenest looking ones. And indeed, they had the highest proportion of green pixels. And UV site reference the lowest. And as my eyes saw, the UV transmitted windows were sort of intermediate. So that was interesting. So it does seem like, yes, the UV radiation, being the one thing that differs between this blue and orange box, does induce a brown coloration. Or at least they are brown, more brown when they're exposed to radiation. But I didn't know if they were more green or more brown, right? So what's the thing changing? I also measured chlorophylls to see if chlorophyll abundance differed in these plants. And here's chlorophyll A, chlorophyll B, and just comparing, if we just focus on the UV filtered and UV transmitted, the paired window design, there's no difference at all in chlorophyll A and a slight difference in chlorophyll B, but in the opposite way than that you might expect for greener plants. The greener plants actually had lower chlorophyll B than the control window plants. So the difference in greenness is not due to chlorophyll content. I can say that. Interestingly, the ratio of chlorophyll A and B does differ between the UV filtered and the UV transmitted plants. There's a higher chlorophyll A B ratio when the UV is removed, which higher chlorophyll A B ratio is a high light response. This is something many plants do when they're exposed to high light. They'll shift the proportion of chlorophyll A and B. So we're seeing a high light response in these plants when we remove UVB radiation. So that was a little bit confusing. In fact, it had me going back and triple, quadruple checking to make sure I didn't mislabel these, because I couldn't, at the time, I didn't really understand, maybe still don't, why you remove ultraviolet radiation and the plants experience it as higher light. But I do have some hypotheses, so we'll put it all together and figure it out. I also compared mRNA transcript abundance in these field manipulated window plants, and there's a lot there. But one of actually two of the top differentially abundant transcripts were for genes involved in flavonoid breakdown. Flavonoids are common UV sunscreen molecules in plants, and they absorb UV radiation. They also act as antioxidants. Um, so it's tricky, but these genes are thought to be involved in flavonoid breakdown. So what we see is more flavonoid breakdown when UV is removed in the UV filtered. And that makes sense if the brown coloration is a flavonoid. There's less of it in these. So the other thing I wanted to do, these plants were in the field dry. I collected them at the end of spring, and they were dry. They spend most of the year dry. And I wanted to compare some indication of their health. A common metric for that is this chlorophyll fluorescence metric, FV over FM. Really, it is a metric of quantum efficiency of photosynthesis. But there is a correlation with sort of stress, or inverse correlation with stress. And the higher it is, the more sort of healthy a plant is, or operating efficiently. It would be the best interpretation. And there's an optimum right around here. No plant can be 100% efficient. There's always some loss to heat. But right around here, 8.84, 84% is sort of perfect plant. And so I took these plants that were dry and hydrated them, kept them hydrated for 8 days, and in very kind of low light, cool conditions, just gave them a very nice, comfy habitat in the growth chamber, and measured their chlorophyll fluorescence, FV over FM, at half an hour after rehydration, and then again 24 hours, and then 8 days later. At that half an hour mark, the 3 treatments were not different at all. The windows and the site reference, no significant difference. And they were very, very low. Normally you see values this low and you should be worried that your plants are about to die. And I was. So I was happy to see the next day they did not die. They recovered a lot. They're not up to that optimum yet, but they are doing really, that was a remarkable recovery for one day. Also, the 3 groups are significantly different from one another. With the UV filtered plants having the lowest FE over FM. That was surprising because I, again, triple checked the way I labeled them. There was no mistake there, but I thought for sure ultraviolet radiation is a stressor. And if you remove that stressor, those plants, I was so confident that those plants would be by far the happiest, the healthiest, because I removed a stressor for a whole year. And I was seeing the opposite. They were the least efficient photosynthesis, maybe the most stressed. Interestingly, is sort of the same pattern with the site reference being the highest and the control window intermediate. I think that what we're seeing here is that the windows did have some effect overall. But again, this blue and orange are only differ in that ultraviolet radiation. So that's the best comparison to look for UV radiation. 8 days later, this pattern continues with the UV filtered plants having the lowest FE over FM and site reference plants having the highest. They're a lot more recovered now, but still that difference persists. So removal of UV radiation, all I can say at this point is that it inhibits recovery from desiccation. I have some other data to kind of figure out why that might be, but that much is clear. Without UV radiation for a whole year, they do not recover as quickly as those that had UV radiation. So that was very surprising to me. I looked at some photosynthetic pigments in these plants, maybe to help explain that color difference, or ideally something about this photosynthesis result I'm seeing. And the ones that had a significant effect I'd like us to focus on here are zeaxanthin and neoxanthin. I mean, in particular, zeaxanthin is higher when UV radiation is removed. Now, this was again another high light response. Zeaxanthin in a typical plant, in a typical setting, will increase when light becomes more intense, when stronger light. Here we took UV radiation away and zeaxanthin went up. That was surprising. Now, a note is that zeaxanthin does a lot of things, accessory pigment, but it is also uniquely associated with sustained non-photochemical quenching, which I'll get back to. But if that means anything to you now, keep that in the back of your head. So another highlight response without UV radiation. But I could answer some questions. Does UV radiation induce brown coloration in Canada nervus? Yes. The coloration is not due to changes in chlorophyll. And how does UV radiation affect recovery from desiccation? It improves it, or at least there is stress when UV is removed, or inhibition of recovery. So that was really a wake-up call. What I learned was I had biases. That greener does not mean healthier, at least not always, at least not in this case. I was so sure that these greener plants were peak health, and they weren't. They were the opposite. So that was interesting. And that stressors are not always stressors, that dosage matters, and that organisms evolved to take advantage of environmental resources. Don't know exactly how they're doing that yet or why, but it seems clear that they have evolved to use this UV radiation in the field, and when they don't have it, they feel the effects of that. And that for myself, I learned that studying plants in nature is a way to fully understand how they interact with their environment. It's very difficult to recreate the sunlight in a lab. It's impossible, I think. The intensity, the frequency, the proportion. And so it was only by doing this in the field that I think I could have gotten these answers the way I did. All right, I'm going to go a little quick because I don't want to run out of time, but just a little bit more I did on stress physiology to try and understand what I was seeing. So I wanted to know, is there variation in response to ultraviolet radiation within Centrichia? We've been talking about Centrichia cananervis. It occurs in a desert habitat, but there's also a close relative, Centrichia ruralis. It's a quite widespread species, that's sort of a species complex, but this particular genotype I'll be talking about occurs in a temperate habitat here from the Bow River in Calgary in Canada, high elevation, cool, relative to the desert, wet, relative to the desert. So these would be my comparison to see if there is some clue in how they're adapted to UV radiation. And so I did this lab experiment. I grew these two in a ultraviolet radiation free environment, just PAR, and then exposed them to moderate levels of UVA and UVB radiation and assessed their transcriptomes at 3 time points before exposure. So they hadn't seen any UV radiation in their life, 10 minutes after exposure and 30 minutes after exposure. First thing to note, here's a PCA of all of the transcripts in the 2 species at the 3 time points. And Canada nervus is here in the pink. First thing to notice is there's a lot more variation between the species. Notice the percentage on PC1 than there is within a species, or a genotype, really. This is one genotype. Super different response in the 2 species. But so I recreated these PCAs looking at each species independently, and you can see a bit more variation is explained by exposure to UV with the 3 time points sort of separating out a bit more, especially in this Centrichia cananervis. It's still not super clear, but that continues to be true. I tried to do this sort of cluster analysis to find clusters of genes whose transcripts respond to UV radiation similarly to each other in each species. And these are some that were notable for their species-specific responses. These are genes that had similar patterns within a species that differed from the other species. Um, what I'd like you to focus on here is these three clusters that have strong Go annotation patterns. These two here were involved, had a lot of genes involved in DNA binding and nucleus kind of orientation, which may be related to DNA repair because DNA absorbs UV radiation. So this could have been something with that. And then these here in membrane, which I maybe a stress response, or I'm not sure really. Oh, oops, went the wrong way. Here are some top 10 most species-specific response genes, and the one of interest here, and remember that pink is Syntrichia cananervis and green is Syntrichia ruralis from the temperate habitat, is this gene, core2 branching acetylglucosaminotransferase family protein. What I know about this is that it's involved in stress tolerance in Arabidopsis thaliana. I don't know exactly how, but stress tolerance in some general sense. And it was interesting because it's constitutively high, or appears to be, in Syntrichia cananervis, the desert species, but it actually decreases with increased ultraviolet radiation in ruralis. I don't know what to make of that, but it was an interesting difference. All I can point to here is that it seems like there could be some constitutive stress tolerance, maybe from the plant that's adapted to desert stressful habitats. Where I started to wonder if there was not some relationship between desiccation tolerance and ultraviolet radiation, actually from that recovery study I talked about. And because I thought, it's what if ultraviolet radiation induces some response that is also beneficial for desiccation tolerance. So I looked at these gene families and like groups of genes with similar predicted functions. And in particular, I would like you to look at these LEA and ELIPS, or late embryogenesis abundant and early light inducible proteins. We know these are super commonly associated with desiccation tolerance. And in Syntrichia ruralis, these had, and this plot shows the log-fold change in abundance. And when the red little, the dots are individual genes, and the red is when they're significantly differentially abundant. But here I'm comparing the whole group. Is the whole group differentially abundant? And in Syntrichia ruralis, they are not. But in Syntrichia cananervis, they are. So as a group, there are a couple individual genes that came up as significant, but as a group, they are also differentially abundant with exposure to ultraviolet radiation. So this was interesting because this could suggest that ultraviolet radiation induces LEA and ELIPS, which could then be useful for desiccation. And if without this cue of ultraviolet radiation, it's possible the plants are less protected or prepared from desiccation. Those plants that were in the field for a year without ultraviolet radiation still had a very stressful and typical desiccation rehydration cycle. And so if they were missing even just a little bit of this extra protection from UV, that may be what we were seeing in the differences. So overall, there is a little transcriptomic overlap in the transcriptomic response between these two species. And so there does seem to be some sort of specific adaptations in the desert plant. And it could be, for this last, I'll be quick, this last one, I wondered if it's not because of when they experience the UV radiation. So in the Mojave Desert, they don't really get, in this area, not a lot of monsoons, they spend the summer dry and the winter wet. So when ultraviolet radiation is strongest in the summer, longer days, less clouds, they're dry and not doing anything. But when they're wet in the winter, they have much lower UV radiation because there's cloud cover when they're alive. On the other hand, in the Centrachia ruralis plant, they are exposed to UV radiation in potentially high amounts because they're high elevation, while hydrated, potentially. It doesn't have this seasonal pattern the way the desert sites do. So I wondered then if UV radiation could be a phenological clue related to seasonal desiccation stress in the desert plants. Okay. So this last quick experiment, I'll try and be quick. I crossed ultraviolet radiation and desiccation stress to see if there's some interaction. I grew these same two genotypes in two light environments, a factorial design, one that had UVB radiation and one that didn't. And then some of them I gave a dehydration stress and some I didn't. And then I compared their transcriptomes and metabolomes. And so this is kind of what it looked like when they were just getting started. And I'll use this dark blue to mean the low UV environment and the light blue to mean the high UV, with hydrated and dry being with or without a drying treatment. Now, what I know going into this is that UV radiation tolerance and desiccation tolerance are correlated in mosses. This has been tested empirically, and they just are. Plants that are tolerant of one tend to be tolerant of the other, but we don't really know why that is. It could be something like this, that there are shared environmental cues that lead to different metabolic responses. So it could be the same UV or drying, it doesn't matter. You'll get both protection from UV tolerance and protection from desiccation. Or it could be something like this, that each stressor, UV radiation and drying, leads to a unique protection that also gives a protection for the other stressor, like a cross tolerance. So this is crosstalk and cross tolerance. Ultimately, they have the sort of same effect, that that there's benefit from having both. So here we're looking at the transcripts in these treatment effects, and on the left, the left is transcripts and the right is metabolites. The species difference is huge again in ultraviolet radiation treatment, pretty small effect. Dehydration had quite a lot. In metabolites, it's a similar pattern, but a little more balanced. In fact, dehydration had the biggest effect on the metabolome, bigger than species. Here I'm going to show the top 5 transcripts that were significant for dehydration stress. And just to explain how this figure will look, if we want to compare the effect of the drying treatment, we look across here. This is Centrachia canadensis on the top row and ruralis on the bottom. And so you could see this particular uncharacterized protein transcript increased with drying. In both light treatments, which are the blues, you can compare just the effect of the light, the UV radiation. This one went down with UVB. And if you want to compare species, you compare this whole top row to the whole top row, or whatever component. And so here are the top 5, a lot to look at. What I want us to look at in particular to start with are these 2, which are ellipses that came up as important for dehydration stress. It wasn't a full desiccation, but they were dry, leaf curling, and I can talk about that later. But these are ellipses, and what's interesting is that as expected, drying induced increased abundance of ellipse, but also so did UVB radiation in almost all situations. This one, it did not, which is interesting too. And this maybe isn't surprising because it turns out some ellipse are reported to be mediated by UVB via the plant UV receptor. UVR8. So maybe that's what's going on here. And it's consistent with the other study I've had. Also, this Leah came up and again increased with drying, we can compare here, but decreased in at least 3 of the 4 cases with UVB radiation. So that was interesting that there seems to be a opposite response, and I don't know what to make of that yet. That is interesting. This project is still a work in progress, by the way. Looking at the metabolome overall, polar metabolites responded to species and UV radiation much better than non-polar. The dark solid line is ruralis, and the dashed line is cananervis. And the triangles are the drying treatment, and the circles are hydrated. One interesting metabolite is this transferulic acid metabolite that increased with UV radiation in Centrichia ruralis. It is a hydroxycinnamic acid that absorbs UVB range comparable to flavonoids. It didn't change with cananervis. So this may be a case of constitutive UV radiation tolerance in cananervis, but a plastic response in ruralis. What I learned here is that, and there's kind of a bit more that I didn't explain, but more crosstalk in cananervis and more cross tolerance in both that supports this concept of UV radiation as a beneficial stress or a use stress that in one way or another helps the plant be tolerant of another stressor, in this case, desiccation. There were other phenolic metabolites and transcripts that I didn't go into because I'm running out of time, induced by ultraviolet radiation that may also provide additional protection from desiccation or vice versa. So I want to say this because I really like it, but it'll be very fast. Where I want to go with this is that this recovery from desiccation showed that they were, over this time period where they were very, very low and then got healthy over 8 days, or quote unquote healthy, was actually a relaxation of a sustained non-photochemical quenching. So they weren't injured in that first time point when their Fe over Fm was very low. They were just quenching in a protective manner all of the light, and it took 8 days for them to relax that process. So we know that much from these plants. And this is a process that has been most studied in an ecological setting anyway, in high elevation conifers. When light is high in the winter, such as here, but temperature is low, perhaps too low to do photosynthesis. Also, water might be in not liquid form. Everwintering overgrains shut down their photosynthesis and do this seasonal quenching. What I think could be happening in Centrichia is that in the summer, sort of analogy, when light is high but water is low, it's possible these plants have a sustained photoprotective quenching for the whole summer. And what I am curious about is whether that is whether UV radiation could be a seasonal cue. It is a seasonal cue in other plants for phenology and other things. And so this could be one way that it tells the plants that it's almost summer, shut everything down, and, buckle in. So I have big plans, to test this. I haven't done any of this yet, but I'm excited about it if you want to talk about it. And then with field and greenhouse experiments. So to wrap, I'm so sorry about going over. This is kind of the research I'm doing. I'm pretty new at all this, but like to acknowledge my collaborators and everyone who helped with this, everyone who funded it. Thank you so much. Again, I'm sorry. Oops, me.
Thank you so much for the great talk. We are a minute or two over, but Jen, I
I'm
think
so sorry.
no, you're totally fine. You have a couple minutes to hang out with us for any
I
questions.
do. Yes.
So So of course, if you need to go, feel free to roll out, but please put any questions in the Q&A chat and we can address those with Jenna. While you all put any questions you may have in that box, I want to go over a couple reminders. But first, I just want to just a moment, round of applause and a big thank you for joining us today. So some reminders. We are hosting seminars the first Thursday of every month this fall and winter. We hope to see you back next month for a presentation on quantum ghost imaging in plants by Duncan Ryan. That's going to be on December 5th. Registration for that's going to be on our website in about a week, so please keep an eye out. If you aren't already, I encourage you to join our mailing list so you can stay in the loop for events or follow us on social media on LinkedIn or Twitter. That's where we are most active. So with those announcements, any questions for Jenna, please put those in the Q&A chat. Okay, first question has just come in. So from Rose Marks, great talk and thank you, Jenna. The UV
Thanks, Rose.
filtering experiment in the field seems to imply some priming effect.
Oops.
Is it possible UV primes plants for desiccation? What are the implications for our laboratory studies that look at response in plant growth in hydrated conditions and no priming and generally low light?
Yeah, that's exactly the challenge, Rose. Thank you. I think that's what's happening. And this is something that's sort of, well, there, I think, similar processes in vascular plants too. I think UV radiation can induce lignification and I protect against drought stress. So I think that's what's happening, but it is tricky to try and study these in a lab setting, even though I do try, but because it's so different from the field, and we're using then plants potentially that are not acclimated to the field to begin with. And the light is so different. UV radiation in the lab tends to be a lot stronger, actually, or a much higher proportion of the light because the PAR is much lower than the sun. And then fundamentally, the plants typically experience these levels of UV while dry, and so you can't grow plants dry. So I don't know the answer, but that it's a challenge that I've been trying to work through with all these different projects, and hopefully again, I would love to hear your thoughts on how to try and tackle that.
Yeah, great. Thanks so much. And Jenna, I think your email's on your lab website. If people want to reach out and follow up about any of these things you talked about.
Oh yeah, definitely. Yeah, it'd be great. Yeah.
Cool. Other questions for Jenna? We have a question coming in from Cara Hunter. So, do you find that the priming effect of UV exposure and desiccation tolerance helps with other stressors as well? For example, freeze-thaw cycles or extreme cold. Does this priming effect happen in other kingdoms of life outside of plants? So, two-part question there.
Yeah, great question. I don't know. I haven't done anything else in these, and I could go back to the literature to look outside of plants in particular. Um, I wouldn't be surprised if it had a benefit to other stressors. A lot of these, another reason it's kind of tricky to tease these apart is because a lot of these antioxidants and pigments and processes to tolerate stress are a little bit generic, like antioxidants that can apply to any stressor that results in reactive oxygen species, which can be a lot of things. So it's hard to, and that's good for the plant because then there's potentially a priming effect for all sorts of stressors. But some of them have dual roles depending where they are or what sort of conformation they're in. Some of them absorb UVB directly, some don't. And so it's hard to tease apart exactly what it's doing and where it is in the plant. So I think one thing I would like to look into is localizing these things and look at changes over shorter time scales to try and get at the mechanism a little more. And I think I love the other stressors thing. A lot of Centrichia live in high elevation and seem to be particularly cold tolerant. They'll photosynthesize under the snow. And so that's one thing I super want to look into. other organisms, I don't know. That's a great question.
Great. A couple other questions have come in. So one from Thomas Boothby, again, a great talk. And apologies if this was missed earlier in your presentation, but do your plants' ability to survive stress diminish if you move them from the field to the lab? If so, have you looked at the transcriptomic or metabolomics of the plant obtained from the field versus the lab?
Yeah, no worries at all. It was a lot, and I was going fast at times. I you can collect from the field and keep them alive in the lab for long enough to do one of these experiments. The 8-day one, probably two, without and the only time I did that was in the Windows study, and I actually didn't bring them in. I did their transcriptomes dry, straight from the field, which is a bit of a snapshot of what's happening right then and there when they dried. So I think there's a lot of potential for that, and something I'd want to do in the slides I blew through at the end of like next steps of figuring this out over a year for many populations and try and really understand what's happening in nature with these plants. So yeah, I think that's totally doable.
Great. And then a question from Olivia Carmo. Again, another thank you for the amazing
Oh
talk.
gosh.
I think Olivia thinks she remembers seeing a slide demonstrating changes in expression of DNA binding proteins between species. Did we see an effect on membrane-associated proteins too? And if so, could it be related to membrane repair following desiccation? So the recovery process.
Yeah, I did. One of the clusters was about membrane. It was Go terms related to localization membrane. I can't remember what genes were in there. And I should note that a lot of these are predicted functions. We do have annotated genomes for these two species, but yeah, a lot of it's predicted functions. But so this result here was a lot of cluster about membranes. And yeah, so I think that I'm not sure what, less familiar with these sort of mechanisms, but in some way, what UV radiation would be doing to sort of, would it be dual protecting against UV and subsequent desiccation, or it can be the cue and the stressor can be totally dislinked if UV radiation and desiccation stress correlate in nature, which they do for these desert plants. They're kind of always together. Then the cue can be UV radiation, and the stressor that it helps could be desiccation. And there is no benefit for UV itself. That's totally possible. So I think I just had a harder time explaining what these would be doing with UV, but it actually makes more sense if I think about it in terms of desiccation.
Great. And then another question from Manny Maran, Balaka Umaran. I apologize if I mispronounced that name. The Leah and ELIP are like intrinsically disordered proteins that are already known to be differentially expressed in anhydrobiotes, correct? And if so, do you come across any other novel proteins?
Oh, yes. Great question. Thank you. I skipped over a lot of the results are uncharacterized proteins, actually, and it can make doing like transcriptomics pretty tricky. I don't know if I put too many of them on here. Oh no, they're not. But yes, definitely I would have a top 10 most significantly differentially abundant, and half of them would be like uncharacterized protein. So there's a lot of potential for figuring out what those do. In a recent project with collaborators, this sort of thing happened with desiccation stress, and where there was a uncharacterized gene, and my collaborators expressed it in Arabidopsis, and it was a transcription factor that regulated drought stress in Arabidopsis. So there's a lot of potential that figure it out, I think, but I haven't done any of that. I don't know where I put it. I'm sorry. Um, but yes, I guess I don't have the names here. Several of these, for example, are uncharacterized proteins. Thank you.
Great. Well, thank you so much. I think that was the last question I'm seeing. We'll see if one more rolls in before I finish talking. But just want to say another round of applause. A big thank you so much for joining us today. And we hope to see you all back at future Wall events. So no more questions have come in, so I think we're okay to roll out. Thanks again, Jenna. We'll see you all
Thank
soon.
you so much. Thank you, everyone. Look forward to chatting more.
Yeah,
Bye.
absolutely. Bye-bye.